Patterson here, and today we're going to go over cell communication, AP Biology Unit 4. So there's different types of communication, and communication between cells has to have a lot of things happening at once. If I am asking you to go and pick up a book, I say go pick up a book. And these sound waves are moving through the air. It's hitting your ears in a very specific pattern, and your brain is going to relay that information, and you are going to know that I need you to pick up the book. And then that will create a response. You'll either pick up the book or you won't pick up the book. And so this is kind of what cells have to do. You have a communication with chemicals or some type of stimulus that's going to cause a cell to respond in a very specific way. So there are three types of cell communication. We have direct contact, and as it states, it's direct contact with it. So if I am actually touching it, then this is going to be a direct contact. So this piece is going to have to come in contact with this. You can see these little knobs are very specific. They're going to be very specific to the other one that's going to open it. So it's going to be a direct contact with a very specific type of cell. So that piece will go in there. It's going to attach, it's called a conformational change, and this will make this part of the cell react, or this cell react in a very specific way. So with direct contact, they're right next to each other. The signals are going to pass through junctions in the cell wall or plasma membrane. So remember, all these cells have some type of casing. They all have a plasma membrane. Plant cells have that cell wall with all that cellulose. The plasma membrane has to allow stuff to go through. So it's going to pass signals through those junctions. So animal cells are going to have what are called gap junctions. So remember, they're not perfectly round, they're different shaped, and they're going to be close enough together where I can create these little pores that will go through, that will allow material to pass from one cell to the next. Plant cells have what are called plasmodesmata, and those are going to be these openings in the cell wall that kind of are like little tubes, little straws, that will allow material to pass back and forth that way. So the next type is local signaling. And if it's local, it means it's nearby. It's not touching, but it's close. If it's locally available, it's going to be pretty close to you. So local signaling is kind of that same thing. So with that, I have a cell. It is going to secrete a type of response, okay, a little signal out. And then the cells around it, the cells nearby, will be affected by it. So one cell secretes a chemical message like a ligand. This is similar to a substrate. So remember, we talked about enzymes. We have a substrate. That substrate goes in and it fits on very specific enzymes. A ligand kind of works the same way. So just think of these as like little pieces that have to go and connect and touch these other cells. That's going to travel through this extracellular fluid. So that's the fluid outside of the cell to another cell. It's going to go a short distance. These will not travel very far. We have chemical messages where we create a response in another cell. So an example of this is paracrine signaling. This is going to do exocytosis. I'm going to have a vesicle with a chemical inside. It's going to come to the membrane. It's going to open up. It's going to allow that chemical to be secreted out of the cell. There will be receptors on other parts of other cells that will take this little piece, it will attach, and it will cause a conformational change and create a change inside the cell. So they'll do that, and then I'll have a response happen due to that connecting of the ligand to the receptor. The other one are synaptic signalings. You see these in neurons. You have a synaptic cleft, which is a space here in between. Again, I have like a vesicle, kind of a chemical signal is going to come through. It's going to leave, it's going to exocytosis, it's going to jump this gap and be attached to the other side to go and continue on its way. But there's a space in between, but they're pretty close together. So paracrine is when I secrete these regulators or these ligands to nearby cells. Exocytosis is going to be that process. And then with synaptic, you're going to find these in nervous systems. So your nerves really don't touch. You have these receptors that are close by that allow stuff to transmit or jump this gap. So they say it secretes neurotransmitters. So this is a neuron response that goes through. So it jumps this gap, or what we call a synaptic cleft. The third type is long distance signaling. And this is going to be where I need to go a very long distance. So I want to be able to travel really far. So to be able to do that, I have to be able to hit our bloodstream and be able to go through that. All the blood in your body goes through your kidneys every 5 minutes. So it's able to get to so many different places very quick. It's the fastest way to kind of get this chemical throughout your body. So I have hormones. Plants have hormones. They're going to release these hormones through their vascular tissue, into their roots, or out through their leaves. They're going to be able to get messages really, really quick. A good example are giraffes. They like the acacia tree, and they will go and eat it. But when the tree starts to be eaten, it creates a panic response and creates like what's called tannin, and it makes the plant very bitter, and it can be very toxic to animals. So in order for giraffes to eat, they're going to eat kind of like upwind from it. So the wind won't pick up and go to the next tree. So if they're in an area where there is wind and it moves, they're going to travel further away outside of that, so the tree won't have that response. So these trees can create a chemical response. It's almost like chemical warfare against things. You also notice this with marigolds. If you ever have marigolds and you mess with them, they kind of release a really weird kind of off-putting smell. And it's another response. It's a chemical response to try not to be eaten. So it can release that, but the whole tree, the whole plant is going to recognize that response and react. Animals will have that with hormones. So hormones, remember, are a type of lipid, which is good because our blood is very aquatic, and so we don't want these hormones to break apart and fall down as they're going through your body. So these hormones are going to be a lipid, so they're going to be hydrophobic. So they're going to move through very, very quick. So our endocrine system has a lot of specialized cells that are going to release hormones into your system. And that way they can reach tissues very, very fast. So if you become scared, adrenaline will pump through your system, and that will be a response to all of your organs to expand blood vessels or other types of responses in order to do that fight or flight response. So there's 3 stages when we do cell signaling. So those other ones were types of communication cells can have. But cells signal in 3 stages. You have a reception, you have a transduction, and you have a response. So this was when I was saying, I need you to get the book. That's me saying, this is what I need you to do. Transduction is going to be my voice going and moving through the air to your ears, that movement from point A to point B. Response is going to be you getting the book. So this is kind of what your cells do. So they're going to have something is going to announce, it's going to be getting there, and then the response is going to be that part. If you just remember this, it will help out quite a bit. So you have reception, you have a target cell. So right here we're going to say this GPCR is my target cell. And I'm going to bring in a signal molecule, and that's my ligand. Remember, those are kind of like those substrates we have with enzymes. So this ligand is going to come in, and again, it's going to fit this spot. And then the receptor is going to interact with this ligand. So this receptor on this target cell is going to react when this ligand attaches. So it's going to go in, it's going to attach, and I have other ones that can come in and they can attach. The ligand and the receptor are going to cause that conformational change. And remember, if you change the shape, you change the function. So that's going to come in, it's going to attach, and now I'm going to have a change in shape. These are going to be temporary. When this leaves, this will go back to being a solid. And so now the receptors can interact with other molecules in the cell. So this is what we say, it initiates the transduction signal. This has come in, this has said, all right, this is what I need you to do, and now you're in the process of getting that done. The receptors can be in the cell itself, so let's say this is inside my cytoplasm inside the cell, this is outside the cell. So this could be a paracrine response. I have something that's been secreted, it's coming in, it's hitting this, creating a response, or I have material inside the cell that is creating a response. So when that happens, I'm going to have a change of shape, change of function. So we have what are called G protein coupled receptors, and these are what's called a GPCR. So this is a GPCR. And what happens is you're going to have a GDP, which is a G protein. This is inactive because it's kind of like ATP and ADP. ADP is energy poor, ATP is energy rich. GDP is energy poor. So I can turn this into GTP. What do you think I need to add to this to make this GTP? I probably need a phosphate. It's going to turn that into a energy rich molecule or a protein. So this can come over here, it will do work, it will grab a phosphate, and then this is going to initiate this and it's going to activate, it's going to turn it on. Because remember, the ATP, your phosphates are like coins. I put coins in, the machine's going to work. I take the coin out, the machine's going to stop. So for me to activate this, I got to put a coin in. So I brought in my ligand, I've activated my GPCR, now I've turned on my conformational change. This GDP is now going to come in. It comes in, it's going to get activated. I got a phosphate that's got put on because now I'm GTP. GTP, and look at my shape. My shape has changed. So I've had a conformational change. And that's going to go now, and that's going to do work someplace else. So this GTP will come in, and this andylate cyclase will be activated, and it can start a response. This will come in, it's kind of like dominoes. I've gone in, I've hit this domino, and that causes this domino to move, and then this domino causes this domino to move, and then this domino causes a reaction, and that's kind of what's happening with these with this reception. All right, so now this has now been activated, and so now I just keep moving down the pathway. I'll get a cellular response, and I can also do it the other way. If I want to take out a cellular response and I want to stop something, I can take it out. I can put it in. So we have what are called ligand-gated ion channels. Remember, ions are charged. So if I want to get material through here, a lot of this material cannot go through this membrane, remember? Only small and nonpolar can go through. Anything that is charged, ionic, or large or polar is going to need a pathway to be able to get through. So I have a ligand. A ligand comes in and activates and opens it up. Now all of this material can go through. I'm going to get this cellular response where material is going to be able to go through this. Now, if I want this to stop, this ligand will go away, conformational change will happen, and now all of a sudden it's closed. So I can deactivate it and I can stop my cellular response. So transduction is the conversion of a signal that will start out outside the cell, go into a signal that goes into the cell that's going to bring about a response. So we have two. We have the signal transduction pathway and a series of molecular changes. Okay, remember, change of shape, change of function. So I'm going to have something attached that will do that. The signal transduction pathway, again, it's like a domino path. It's going to keep going and knocking the next domino to get you to the end of that path. So messages can be phosphorylated by the enzyme protein kinase. If something is phosphorylated, what is happening? All right, a phosphate is being added to it. Okay, so I'm being phosphorylated. So protein kinase is going to bring in phosphates. It turns on the signal. Okay, so this is going to turn it on. Because remember the coin, I have to add that coin for the machine to turn on. So here I have a protein. Protein kinase is going to come in. I'm going to take ATP to ADP, and now I have added this phosphate to this protein. I have changed the shape. I have turned on that signal. So now I'm activated. When I have messages become dephosphorylated, D means to not, so it's going to take away a phosphate by the enzyme protein phosphatase. So phosphatase is going to be the off button. I take out that coin. If I take out the coin, what happens to my machine? It stops. So when I take out that phosphate, now the signal's blocked, it's shut off, and my signal starts to slow down. So here I have protein, I have a phosphate, that protein phosphatase. I'm going to dephosphorylate, I'm taking off that phosphate, and now I just become a regular protein and I'm deactivated. I'm shutting down my signal, I'm slowing it down. We have secondary messengers. These are non-protein, they're small, they're water soluble, and they're ions, so they're charged. These help move a message by amplifying the response. So it's kind of like the 12 Days of Christmas song. I start with 1, I have 2, I have 3, I have 4, and it kind of keeps going and going and going. So I have one message, okay, and then I get to 2, and then I'll get to 4, and then I'll get to 8, and then I get to 16, and so it kind of amplifies in what's called a cascade. It's kind of going to go small and it's going to go out. So these will amplify the response. We call this CAMP. It's a cyclic AMP. This is like ATP. So remember, ATP is adenine triphosphate. I have 3. And ADP is adenine diphosphate. When I have AMP, is adenine monophosphate. So how many phosphates do you think CAMP has? Has 1. So it's gotten rid of 2 phosphates. And if I've got rid of 2 coins, I can do a lot of work with 2 coins. So those 2 phosphates are going to go out and they're going to help get messages turned on and go wherever they need to go. So this is a good way to amplify. I'm multiplying my responses out to get more of a response to occur. So I have, again, my GPCR. My GPCR is here. It's activated. I have my ligand on it. My GTP is over here. ATP has come out. So my 2 phosphates are there. So now I have CAMP, okay? And so now these 2 phosphates will go and do work, okay? So it's a phosphorylation cascade. So when I have this cascade effect, I have CAMP comes in, and the phosphate is going to react with a molecule. So we're going to have it react with this one. This is going to cause a reaction that will happen with this molecule. And now I have a phosphate here. I've turned on this signal. And then I'll have this will come in. Okay, this will come in and it will do the same thing. I'm going to bring in and I'm going to create another signal here. And then it's going to keep doing this until I get to my target protein, my target part of the cell. Wherever I need to go to get that response to happen, that's where I'm going to go. This is going to be an exponential growth. So I start out with, again, 1, I go to 2, I go to 4, I go to 8, I go to 16. Okay, so it's going to start spreading out bigger and bigger. So the response with the target cell is going to be to the signal molecule. So I have to have that signal to make that target cell respond. It's going to convert that signal into a response that will either alter or modify a cellular process. So a good example is insulin. I eat candy and my body has to respond to that sugar increase. There's going to be an alert or some type of signal that is going to say, I need more insulin to break down all the sugar. But then when you get to a point where your body's broken down enough sugar, you have to signal that insulin to stop. So you have like a feedback mechanism where you're turning on and turning off. So you have a fine line you have to draw when you're kind of working these two things back and forth. So here I have my GPCR. It gets activated, GTP. It's going to cause this to create a response. And then I'm going to create CAMP, and that's going to turn on this part here. But you see these arrows? This means I'm responding. I'm going that way. You see these ones that look like a T? These are inhibitors. These are going to be ones that I need to turn this off. When I make enough of a certain type of response or a certain type of chemical or molecule, I can have a response come in that will actually start to dephosphorylate. It's going to start to shut off some of these molecules to keep them from making more. So this is going to come in, shut this down, and if it shuts this down, then it's going to shut down CAMP. It's going to shut down the pKa, and so this is going to be the response that turns it off. So you have this back and forth feedback mechanism that's going to help modify that cell response. So let's look at an example. Okay, how does fruit ripen? If you have fruit that is not ripe, you can put it in a brown paper bag and close the lid, and it will ripen faster. And so now the question is, well, why? Why does that happen? Well, you have ethylene that gets produced by the plant that causes the plant to ripen. So you have these receptors in the fruit. So it attaches to these receptors, so this ethylene comes into the fruit, and it's going to come here to this ER, the rough ER, and it's going to attach to these ribosomes, and it's going to make a certain type of molecule. And the molecule will go to the DNA, and then the DNA is going to make a certain type of code for protein production, and then that will go and that will help ripen the fruit. So this is a signaling change. This comes in, causes a reaction, causes another reaction, and causes another reaction. So this is your chain reaction. And then your response is going to be the fruit ripens. Now, what happens if you leave that fruit in your bag for 2 weeks? You're going to come back, it's going to be a mushy mess. Okay, this ethylene is staying on, and you don't have the receptors turning it off. So changes in a response pathway, I can change responses with a membrane permeability. If I can make some of these molecules go through faster, go through slower, I can change my response. So I have changes in metabolism. This is going to be how fast things break down and come together. If I change that, I can change my response. I'm breaking down enzymes faster. I'm putting things together faster. If I do that, I can change my response. And then the last one is gene expression. This is going to be in your DNA. For me to make mRNA to go do protein synthesis, I can change that. If my gene gets mutated or changed, I can also change the way my enzymes and the molecules that I create can modify that response. All right, apoptosis. This is basically cellicide. This is when a cell is going to destroy itself on purpose. So it's a program. So it's a program of controlled cell destruction. It is going to recognize that it is not able to function like it needs to. It will send out signals to the cell saying that it's time to break it down and to destroy itself. It does it to protect nearby cells from enzymes that would leak. You would not want your lysosomes, for example, to bust open in a very pH-friendly environment to it. And now all of a sudden I'm destroying everything. You don't want that to happen. And so it's going to break itself down in a very controlled way. It's going to signal that cascade effect where now I'm going to have a signal come in, and then all of these molecules are going to start fanning out and going and starting to turn on all of these specific cells that are molecules that are going to come in and start to say, all right, we need to start shutting this down. We're going to recycle this stuff. We're pulling things out in order for that cell to be destroyed. And this is needed for normal development. When you see hands of a fetus, okay, you can see the hands is kind of webbed and they're kind of like real short here. Well, you need this process to go in and destroy some of those cells so you don't have the webbed fingers like when you're born. You don't have that. So apoptosis is not a bad thing. You need that to replace and to remove certain types of cells that are no longer needed. All right, that's cell communication. I hope that was helpful. If so, let me know. We'll talk to you guys later.